Hello everyone, welcome back to our YouTube channel, Dairy Pathshala, your one-stop solution for all the subjects related to dairy science and technology. So with this video, we are starting a lecture series on dairy microbiology, and today we are going to discuss about history of microbiology. Many of you might be thinking that why the hell we are studying history, right? We are here to study about dairy microbiology, so why we are studying history? So I would say that learning history of anything, regardless of the subject, it's very important to know about the contributions of the great scientists which we have given in the development of the science. And by learning the history, we can learn from our past so that we can apply those learning in our present and future. So with this, we'll begin our today's video. So in this video, we are going to discuss about microbiology as a science. Then we'll be seeing the cell theory. Moving on with the discovery of microorganisms, how the microorganisms were discovered, and then in the next video, we will be discussing about the theories of spontaneous generation and biogenesis. So, introduction to microbiology. Microbiology as a science dates back to 17th century. So, way back in the 17th century, scientists started learning about this science. They started researching and experimenting on different topics related to microbiology. So, as per the definition goes, microbiology is nothing but a science in which we study about microscopic organisms like bacteria, viruses, fungi, and protozoa. And what are these microscopic organisms? These are the organisms which we cannot see with our naked eyes, and we require some sophisticated instrument like microscopes if we want to see them. And whatever the early discoveries which were happened in those times were very foundational to all biological sciences. And in those days, we have not divided microbiology into different branches like we see today as dairy microbiology or food microbiology, but since nowadays, because of the contributions of the scientists, we are so developed, so we have divided microbiology in different branches. And in our channel, we are going to discuss specifically about dairy microbiology, which deals with the microorganisms affecting the dairy products. So that is how microbiology was introduced in the 17th century. So now we'll be discussing about cell theory. But before this cell theory, let's hear some facts about the cell. So the term cell was first coined by the scientist Robert Hooke. So what he did, he used a small and simple microscope or a magnifying glass of 20 times magnification to observe a cork cell. Cork cell is nothing but a material taken from the cork oak tree. So in the year 1665, he observed the cork cell through his magnifying glass, and he observed some structures like small rooms or little boxes, which he first described as cellulae, and later he termed it as cells. His discovery led to the formulation of the cell theory. So on the basis of his discovery about the cell theory was developed. So this is how the Hooke's microscope looks like, and you can observe in this image that the slice of cork, and these are what he termed as cells, these tiny little structures, some square shaped, some rectangular shaped, some irregular shaped, some hexagonal shaped. So these are look like small rooms, right? So that is why he termed these structures as cells. Since this structure was discovered, but there was no definition given by Robert Hooke regarding cell. So to define the cell in proper way, two German scientists, Mr. Matthias Leiden and Theodor Schwann, they given a cell theory. And according to them, cells are the basic structural and functional unit of life. Now you may say that how you can say that cells are basic structural and functional unit of life? Why they are structural unit of life? Because if you divide a human body, you can find different organ systems, right? The reproduction system, the digestive system, the respiratory systems, and these organ systems are made of different organs, right? And again, if you see inside these organs are made of tissues, right? Tissues are combined together to form an organ, right? And if you break down this tissue, you will get to the smallest structural unit that is called a cell. So these cells are clubbed together to form tissues. This is the example in the case of multicellular species like us. like animals or plants, which possess multiple cells. In the unicellular organisms, like microorganisms, which are bacteria or fungi, the single cell defines the whole structure of that organism. In the similar manner, these cells perform all the basic functions which are necessary for a living organism. They perform all the metabolic processes. Whatever the chemical reactions of living cells, this all takes place within the cells. For example, taking up of nutrients, processing the nutrients to generate energy, excreting the waste to originate new cells. So all the cells originate from pre-existing cells. So they do reproduce. That cells contains hereditary information in the form of DNA, which is passed from one generation to another. In the same time, when cells were discovered, microorganisms were also discovered. Way back in the 17th century, in the 1665 year, which we have discussed earlier in the video, cells were discovered. So there was a Dutch scientist whose name is Antonie van Leeuwenhoek. He was very fond of microscopes, and, and because of his curiosity, he tried and experimented and made over 500 microscopes with different magnifying powers. By using these microscopes, he observed microscopic swarms from different specimens, for example, tooth plaque, rainwater, and diarrheal fecal. And by doing this, he became the first person to use microscope to observe microbes. And by doing this, he discovered bacteria, protozoa, sperm cell, and blood cells. And this discovery led to the formation of a new term called as animalcules. Now, what is this animalcules? We are discussing about the discovery of microorganisms, right? Now, what is this animalcules? So, microorganism, this term was discovered later, but before that, he used to call them as animalcules, which literal meaning is a tiny moving animal. Because when he observed these specimens, he found the organism moving. So he called them as animalcules. And later, the term microorganisms was introduced. Because of his contributions and discovery of microorganisms, he is known as the father of microbiology. If any one of you have observed the modern microscope, or which we call as compound microscope, this is how the microscope of Antonie van Leeuwenhoek looks like. So now you can imagine, if you compare it from modern microscope, how we have grown from 17th century to 21st century, how much we have developed. That is how the old microscope looks like, which was used by Antonie van Leeuwenhoek in the 17th century, and how our today's modern microscope looks like. It is interesting, right? To know that how much we have taken a leap in the small span of only 300 to 400 years. So with this, we'll wrap up this video, and in the next video, we will be discussing about the theories of spontaneous generation and theory of biogenesis, and we'll be discussing that among those two theories, at last, which theory wins. And we'll also be discussing the contributions of some important scientists. So till then, goodbye, and do not forget to like, share, and subscribe, and also comment down in the comment section if you have any doubts related to today's video. So see you in the next video.